I hope you have seen my introduction to nucleic acids DNA and RNA. In this video, I will focus on the building blocks of DNA and RNA, the nucleotides. Zooming in on the DNA by itself, we would see the DNA strands twist in a double helix structure. Zooming down further, we would see the individual nucleotides that form the double helix structure. The ribophosphates form the rails of the helical ladder, the nucleobases. A, G, C, T form the rungs of the helical ladder. The four nucleobases of DNA are adenine A, guanine G, cytosine C, and thymine T. RNA has the same nucleobases except uracil U takes the place of thymine T. These nucleobases are joined to a ribose sugar to form a nucleoside. RNA uses ribose for its sugar. DNA uses deoxyribose for its sugar. Deoxyribose is just ribose stripped of an oxygen from its hydroxyl group OH at the 2' carbon location. Without the oxygen at 2' carbon, deoxyribose is less chemically reactive, and so more stable, which makes DNA more stable than RNA. Generally, the ribose is formed with a phosphate group already attached. The nucleobase need only join with the ribophosphate to become a fully fledged nucleotide. Without the phosphate in place, the ribose sugar plus nucleobase is called a nucleoside. The RNA nucleosides are adenosine A, guanosine G, cytosine C, and uridine U. The DNA nucleosides are made with deoxyribose in place of ribose, and use the same RNA names with a deoxy prefix before the nucleoside name. The DNA nucleosides are deoxyadenosine A, deoxyguanosine G, deoxycytosine C, and deoxythymidine T. The addition of one phosphate to any of these nucleosides will make a nucleotide. A nucleotide could also be called a nucleoside monophosphate, such as adenosine monophosphate AMP. With two phosphates added, a nucleoside becomes a nucleoside diphosphate, such as adenosine diphosphate ADP. A nucleoside with 3 phosphates is a nucleoside triphosphate. We should all be familiar with the high-energy adenosine triphosphate ATP, which serves as the energy source for most bioreactions. There are also highly energetic forms of the other nucleoside triphosphates, including guanosine triphosphate GTP made during the Calvin cycle of photosynthesis. Nucleosides and nucleotides also serve as the basis chemicals of many of the metabolic coenzymes, such as coenzyme A, NAD+, NADP+, FAD, and FMN. Nucleotides are further classified by how many carbon rings in their nucleobases. Purines have 2 carbon rings. Pyrimidines have 1 carbon ring in their nucleobase. Adenosine and guanosine have 2 carbon rings, so they are purines. Cytidine, thymidine, and uridine have only 1 carbon ring in their nucleobases. As we shall see, when DNA or RNA molecules are assembled, the purines are always coupled with pyrimidines. Specifically, guanosine G is always paired with cytidine C, so forms a CG pair. Adenosine A is always paired with thymidine T in DNA, and so forms an AT pair. In RNA, adenosine A is always paired with uridine U. From the figures, we can see that thymidine molecule is very similar to the uridine molecule, with the only difference in the base is that uridine lacks the methyl group CH3 on its nucleobase. The RNA nucleotide ribose could be altered into DNA deoxyribose by removing an oxygen from the OH group at the 2' carbon by means of the enzyme ribonucleotide reductionase. The figure shows a side-by-side comparison of ribose and deoxyribose in representative nucleotides. The deoxyribose is simply a beta-D ribofuranose with an oxygen O removed from the hydroxyl OH at the 2' carbon position. The oxygen removal is done by the enzyme ribonucleotide reductase. The construction or synthesizing of any one of these nucleotides is a multi-step process involving many enzymes, amino acids, and energy from ATP. The starting point for pyrimidine synthesis is the amino acid glutamate and carbonic acid. Glutamate is the most abundant amino acid in your blood. Glutamate is synthesized in many organs in your body, including muscle, or could be absorbed into the body from eating meat, an animal muscle. Carbonic acid H2CO3 is simply carbon dioxide CO2 dissolved in water. When carbonic acid loses a proton H+, it becomes the bicarbonate ion HCO3 negative. So, to synthesize the pyrimidine nucleotide uridine, for step 1, the enzyme carbamoyl phosphate synthase uses 2 ATPs to combine glutamine and bicarbonate into carbamoyl phosphate. For step 2, the enzyme aspartate transcarbamoyl ACE adds aspartate to carbamoyl phosphate to make carbamoyl aspartate. For step 3, the enzyme dehydratase removes a water H2O from carbamoyl phosphate to close the ring and make the 6-carbon ring molecule orotate. For step 4, the enzyme orotate PR transferase adds the pentose sugar PRPP that is used to make ribose to the orotate. 1-phosphate P is lost, and the result is orotidine monophosphate OMP. For step 5, the enzyme UMP synthase removes a carbon dioxide CO2 from orotidine monophosphate OMP to leave uridine monophosphate UMP. In the figure, the nucleotide shown is uridine U, which is one of the RNA nucleotides. So, in 5 steps, and using 5 enzymes and 2 ATPs, the common amino acid glutamine and carbonic acid and the pentose sugar PRPP become an RNA nucleotide. All these enzyme-driven reactions occur inside a living organism. I don't know how nucleotides spontaneously assemble outside of a living organism without enzymes or the pre-ribose sugar PRPP. What came first, nucleotides or life? That is a limit of scientific knowledge. Starting from uridine monophosphate UMP, it is just a few easy steps to get the pyrimidine nucleotides thymidine monophosphate DTMP or cytidine monophosphate CMP. To get cytidine C from uridine U, in step 1, the enzyme nucleoside diphosphate kinase adds a pyrophosphate or diphosphate PP onto uridine monophosphate UMP to get uridine triphosphate UTP. In step 2, the enzyme CTP synthase adds glutamine to uridine triphosphate UTP to convert it into cytidine triphosphate CTP. In step 3, the enzyme CMP synthase removes 2 phosphates PP from cytidine triphosphate CTP to leave cytidine monophosphate CMP. The CMP is the cytidine C nucleotide used in RNA. To get the deoxycytidine C used in DNA, we go to step 4, where the enzyme ribonucleotide reductase removes an oxygen from the ribose to convert it to deoxyribose and adds a phosphate. And the cytidine monophosphate CMP becomes deoxycytidine diphosphate DCDP. In step 5, the enzyme DCMP synthase removes the phosphate, and deoxycytidine diphosphate DCDP becomes deoxycytidine monophosphate DCMP, and DCMP is the cytidine C nucleotide used in DNA. To get thymidine T from uridine U, in step 1, the enzyme ribonucleotide reductase removes an oxygen from the ribose to convert it to deoxyribose, and adds a phosphate P onto uridine monophosphate UMP to get deoxyuridine diphosphate DUDP. In step 2, the enzyme DUMP synthase removes the second phosphate from deoxyuridine diphosphate DUDP to leave deoxyuridine monophosphate DUMP. In step 3, the enzyme thymidylate synthase extracts a methyl group CH3 and adds it to deoxyuridine monophosphate DUMP to convert it into deoxythymidine monophosphate DTMP, which is the thymine T nucleotide used in the DNA molecule. On to purine nucleotide synthesis. To make the purine nucleotides, guanine and adenosine, we start with inosine or inosine monophosphate, IMP. There are at least 10 steps to make inosine IMP, starting from ribose 5-phosphate R5P. The R5P could come from the Calvin cycle of photosynthesis. I show the pathway in the figure without verbally going through the steps from ribose 5-phosphate R5P to inosine monophosphate IMP. The basic components of inosine monophosphate are bicarbonate ch03- and the amino acids glutamine, formate, glycine, aspartate, along with the pre-ribose pentose sugar PRPP. To synthesize guanosine from inosine, first assemble inosine monophosphate IMP from its basic components in a 10+ step process. From inosine monophosphate. Step 1. The enzyme inosine dehydrogenase uses NAD+ to split water H2O. 1 H bonds with NAD+ to become NADH. The remaining hydroxyl OH group bonds onto the inosine monophosphate IMP to convert it into hypoxanthine monophosphate. In step 2, the enzyme GMP synthase knocks off the hydroxyl group OH and adds an amino group NH3 by taking the NH3 from glutamine to make glutamate. The result is that hypoxanthine monophosphate becomes guanosine monophosphate GMP. The guanosine monophosphate GMP is the form of guanosine G nucleotide used in RNA. To get the DNA form of guanosine G, in step 3, the enzyme ribonucleotide reductase removes an oxygen from the ribose sugar and adds a phosphate. Guanosine monophosphate GMP becomes deoxyguanosine diphosphate DGDP. For step 4, the enzyme DGMP synthase removes the second phosphate from the deoxyguanosine diphosphate DGDP to turn it into deoxyguanosine monophosphate DGMP, which is the form of guanosine G nucleotide used in DNA. Last, but not least, adenosine can be synthesized from inosine. First assemble inosine monophosphate IMP from its basic components in a 10+ step process. From inosine monophosphate. Step 1. The enzyme adenyl succinate synthase detaches an oxygen O from the purine ring, then attaches an aspartate amino acid at its amino group NH3. The inosine monophosphate IMP is converted into adenyl succinate. In step 2, the enzyme adenyl succinate lyase cuts off a fumarate from the aspartate and leaves the amino group NH3 attached to the purine ring. The result is adenyl succinate becomes adenosine monophosphate AMP. The adenosine monophosphate AMP is the adenosine A nucleotide used in RNA. To get the DNA form of adenosine A, in step 3, the enzyme ribonucleotide reductase removes an oxygen from the ribose sugar and adds a phosphate. Adenosine monophosphate AMP becomes deoxyadenosine diphosphate DADP. For step 4, the enzyme DAMP synthase removes the second phosphate from the deoxyadenosine diphosphate DADP to turn it into deoxyadenosine monophosphate DAMP, which is the form of adenosine A nucleotide used in DNA. For you aspiring medical doctors, genetic scientists, and biochemists, the Ninja Nerd does a detailed professional job of explaining how the pyrimidine and purine nucleotides are synthesized. On his YouTube channel Ninja Nerd episode, Nucleotide Synthesis or Purine and Pyrimidine Synthesis, at the link shown. I went through the exercise of showing how all 5 nucleotides are made, to demonstrate the complexity of the formation of these nucleotides, and to demonstrate the utter dependence on enzymes to catalyze the nucleotide synthesis reactions. My purpose is to cause you to ask the question, How did the earliest life construct its own RNA and DNA nucleotides without the benefit of catalysts? There are at least 5 steps to form the simplest pyrimidine RNA nucleotide, uridine U, from basic amino acids. There are at least 10 steps to making inosine monophosphate from basic amino acids, plus another 3 steps from inosine to get the purine RNA nucleotide, guanosine G. It seems the 13 steps to making RNA nucleotides would be impossible without enzymes. Did the enzymes come first? Many enzymes are complex proteins. There are far more chemical reactions involved in constructing a 100 or 1,000 amino acid protein enzyme than forming a nucleotide. These are limits on our knowledge. Biochemists and geneticists are working every day to expand the limits of our knowledge.
Thank you for watching. If you enjoyed this video and want to help me make similar content, please like and subscribe.